I'm Cameria, and I'm in Miss Boswell's microbiology lab class, and today I'm going to talk to you about gram staining. So first I'm going to tell you the difference between gram-positive and gram-negative cell walls. Gram-positive cell walls have a thick layer of peptidoglycan, and they also have a unique chemical called teichoic. On the other hand, gram-negative has a thin layer of the peptidoglycan. Also, with gram-positive, they have a crystal violet dye in their cell wall that is used for gram staining procedure. Lipoteichoic acids anchor the cell wall to the cytoplasmic membrane. Gram-negative cell walls are composed of liposaccharides, phospholipids, and proteins. Gram staining has a special procedure that you have to follow in order to do it correctly. Gram staining was developed by Christian Gram in 1884. The technique consists of staining microbial samples with different series of dyes. This would leave some microbes purple, which is gram positive, and some microbes pink, which is gram negative. So first you would spread the culture in a thin film layer on the slide. Then second, you would let it air dry. Then the last step is you would pass the slide through a flame to fix it. So this last step helps so that the sample sticks to the glass slide and it doesn't wash off during staining. The first step to gram staining procedure is the slide is flooded with crystal violet for 1 minute and then it's rinsed off with water. The second step is the slide is then flooded with iodine for 1 minute, and then it's rinsed off with water. The third step is the slide is then flooded with a solution for 10 to 30 seconds, and then it's rinsed off with water. The last step is the slide is then flooded with safranin for 1 minute, and then it's rinsed off with water and blotted dry. So, how does each component work? Iodine is a moderate that binds the dye and it makes it less soluble. Crystal violet is a primary stain and it colors all the cells. Ethanol and acetane solution acts as a decolorizing agent. Safranin is the red counter stain that provides a contrasting color to the primary stain. How is gram stain used clinically to help identify the cause of infection? Gram stains help distinguish between gram-positive and gram-negative cells. This aids in identifying the species or narrowing down the possibilities. Another way that gram staining helps after you identified if it's gram-positive or gram-negative, this helps you figure out which bacteria you have and which antibiotic will help cure that particular bacteria.